Hey. Okay, thank you, Andrea, for the introduction, and I thank Limno Seminar Organizing Committee for inviting me to give this webinar, and thank you all for tuning in today. So, as Andrea mentioned, I've been a bit distant from zooplankton work over the past couple of years, as my postdoc work is mostly related to fish ecology. So, putting together this presentation was a good refresher for me. So, I thought of sharing some background in this topic and share some of my past work, and also talk about some of the concerns, challenges, and opportunities. Okay, so I'm joining you today from Regina, which is located on the traditional lands referred to as Treaty 4 territory. These are territories of Nihuak, Anishinaabe Pike, Dakota, Lakota, and Nakota, and the homeland of Métis Nation. Today, these lands continue to be a shared territory of many diverse peoples from near and far, and we are grateful for the opportunity to live and work on this land. I also have a long list of people and organizations that I'd like to thank who supported me during the work I'm presenting today. So this includes my advisors, collaborators, colleagues, and my funding sources. So we know that lake ecosystems are globally threatened by a number of natural and anthropogenic stresses. So this include pollution, climate change, invasive species, dam constructions, and list goes on. So these stresses can influence biological properties of lakes through complex pathways. So they can directly influence biological properties or indirectly through their effects on water quality and quantity of lakes. They can also influence catchments, which in turn influence water quality and quantity, again influencing biological properties. Therefore, we live in a time that we need appropriate biological indicators to measure and track these impacts. So I don't think this group need a detailed introduction into zooplankton. So today I'll be mostly focusing on two groups among major zooplankton groups, so copepods and cladoceraans, because my work was mostly related to these two groups. So use of zooplankton as indicators has a long history. So one of the well-known example is they are used to assess effects of industrial acidification in lakes in Eastern North America and Scandinavia, and to assess subsequent recovery. So most of these works come from Sudbury and nearby regions in Ontario. So another good example is they are used to assess effects of eutrophication of lakes in Europe and subsequent recovery after various interventions. A more recent example is bioassessments in lakes in Western Canada to assess potential impacts of atmospheric emissions from oil sands operations. And this is where I was involved in. So we can think of two major approaches of collecting zooplankton samples. So one is by collecting and analyzing contemporary water column zooplankton samples. Other one is by examining sediment samples for chitinous remains of zooplankton. So each of them have their own advantages and disadvantages. So contemporary sample provide a snapshot of the pelagic community at a given time, unless we specifically sample other habitats. On the other hand, sediments are integrated sample from pelagic, littoral, and benthic habitats. They are also temporally integrated through several seasons or years. However, sediments only provide us information on species that are producing chitinous remains, that is most Cladocera. On the other hand, contemporary samples include copepods, rotifers, and other taxa in addition to Cladoceraans. So both of these approaches can be used for both spatial and temporal assessment. However, sediment samples are often used to assess more long-term changes over centuries to millennia. We can think of number of indicator metrics related to zooplankton that we use to assess environmental changes. For example, we can look at changes in species composition. This includes both taxonomic and functional diversity. Use of individual indicator species is another approach. Changes in body size of individuals and total zooplankton biomass can also reflect environmental changes. Several new studies explored the potential of using stable isotopes and other biochemical material of zooplankton as ecological indicators. So today I'll be only focusing on species composition and individual indicator species. Zooplankton are good indicators due to a number of reasons. They are fairly easy to sample because they occur in high densities and more homogeneously distributed in lakes. Therefore, they are more cost effective than monitoring for fish or benthos. They also have a high taxonomic diversity, and different species have different sensitivities to different environmental conditions. Therefore, the changes in environmental conditions can make substantial changes in community structure. And most species have short generation times. Therefore, changes in recruitment success due to environmental changes will be rapidly expressed in terms of changes in population choices and community structure. Another important reason is the intermediate position in aquatic food webs, sandwiched between these predators such as fish and primary producers. Therefore, they are vulnerable to not only to direct effects of changes in environmental conditions, also to indirect effect cascading to food webs. Sometimes I get this question, why we should even use biological indicators, because it's easier to sample for and use chemical indicators. So one reason is, of course, we are interested in biological impacts of these chemical changes. The other reason is they provide insights into different impact mechanisms of these chemical changes, as we can see here. So today, I'll be sharing some of my work looking at zooplankton communities in boreal lakes across northern parts of Saskatchewan and Alberta. Two of them are broad-scale spatial assessments, and another one is a temporal assessment or a paleolimnological study. So motivation for these studies comes from evaluating the effects of oil sands industry in Alberta on lake ecosystems in the region. So we know that oil sands production has been increasing rapidly since 1980s. I think the production is somewhat plateaued in recent years. However, due to this unprecedented scale of production, there are several concerns about potential environmental impacts. One such concern is the effects of sulfur and nitrogen oxide emissions on lake ecosystems in northeast Alberta and northwest Saskatchewan. So there are two main concerns. Deposition of these acidifying precursors can lead to acidification of lake ecosystems. In addition, nitrogen deposition can lead to increase in lake productivity. So first, I'm going to talk about the spatial assessment we conducted in northwest Saskatchewan. So downwind region in northern Saskatchewan are highly vulnerable to acidification due to their high geological acid sensitivity. In fact, a recent study showed that this is one of the most acid sensitive regions in Canada. Also, at the time that oil sand industry was booming, there were some considerations about expanding oil sand activity to some parts in northern Saskatchewan. However, at that time, we did not know much about the lake ecosystems out there because these are very remote systems. Provincial government initiated the lake assessment project in this region in 2007 to see if we see any signs of acidification and also to establish regional baselines. So I got the opportunity to involve in this project by conducting zooplankton analysis component as my master's research project. So when we look at pH of these lakes, we can see that they are mostly in near neutral ranges, and also sulfate and nitrate concentrations are quite low. In addition, calcium, which is the main buffering cation in these systems, is also low. So this figure shows the acid neutralizing capacity, or the ANC, of our study lakes in the region. So ANC is an index of aquatic ecosystem quality for biota in relation to acid stress. It's also a measure of sensitivity to acidification. So it's calculated as the difference between inorganic cation and anion concentrations while correcting to free organic anions. So in general, ANC below 200 are considered to be sensitive to acidification, and those below 50 are considered to be highly sensitive. So we can see that most of the lakes in the region are sensitive to acidification, while 8% of them were highly sensitive. So I should note here that this high acid sensitivity in these lakes was due to low base cation concentration rather than due to high anion concentration. Also, we did not have any prior data on zooplankton communities from these lakes. Therefore, we thought of comparing our data with other boreal regions in Canada. So this table, we have compared crustacean zooplankton species richness, and the first column represent our study lakes, and these green and orange columns represent acidified and non-acidified systems elsewhere. So when we look at the average of species richness, we can see that it is quite low compared in acidified systems compared to non-acidified systems. So this is one of the most prominent responses to acidification. And we can see that the richness in our study lakes are more comparable to acidified systems rather than to non-acidified systems. We can also see the same pattern respect to range and percentage of lakes with richness less than 5. Then we also compared frequency of occurrences of species with others. So here I have highlighted the acid sensitive species. So we can again see that our lakes are more comparable to acidified systems that are indicated in green rather than to non-acidified systems in Ontario. But when we look at the acid tolerant species, we can see that they are more or less comparable across all regions. And this makes sense because these are generally ubiquitous in most lakes, and we only see an increase in their abundance with acidification. So what these comparisons basically telling us is, although we don't see any widespread acidification, zooplankton communities in our study region are more comparable to acidified systems in Ontario. So we need to think about regional differences here. So yes, all of them are boreal systems, but our study region is located at a higher latitude. So with the increase in latitude, species richness can be low due to harsh environmental conditions and reduction in environmental energy availability. So this highlights the importance of having regional baselines to evaluate future impacts of industrial deposition rather than using data from other regions as a baseline. Then we evaluated what environmental factors were structuring zooplankton communities in our study lakes. For this, we used a method called redundancy analysis. It is a multivariate method commonly used to analyze community data. For those who are not familiar with this method, first, this method summarizes the major patterns in species along different ordination axes. Then it identifies significant environmental factors explaining these species patterns. So here I have summarized the major findings of this analysis. So we can see that along axis 1, two large-bodied species, that is Daphnia pulex and Agrodaptomus leptopus, is negatively correlated to a small-bodied species called Bosmina longirostris. And this is a classic relationship in zooplankton ecology. And this negative association was influenced by fish predation regime, nutrient concentrations, and DOC concentrations. So these relationships make sense in terms of what we know about the ecological affinity of these species from the literature. Along axis 2, we see another negative association between acid tolerant species called Leptodiaptomys moentus and several acid sensitive species. And this variation was explained by A and C gradient. So this is another way to present the same information. So here we are looking at comparing relative abundances of L minutus between different acid sensitive categories based on A and C. And we are also comparing this abundances between acidified and non-acidified systems in Ontario. So we can see that abundances increase with increase in acid sensitivity. And we can also see the same pattern between acidified and non-acidified systems in Ontario. And we see the opposite pattern for acid sensitive species, which again we see is similar to difference between acidified and non-acidified systems in Ontario. So one interesting thing here is that these large-bodied species seems to be uninfluenced by A and C gradient. So this is maybe because these species are pre-adapted to natural acidity caused by organic acids in these bog-type lakes. So along axis 3, we see another negative species association, and it was related to nutrient concentrations. And again, this was in agreement with what we know about the ecology of these species from the literature. So overall, we saw significant correlations between zooplankton community composition and two main environmental gradients that are vulnerable to industrial deposition, as named acid-base status and productivity. So this suggests that we can use zooplankton communities to track environmental changes in this region in the future. However, it is important to consider regional differences when we are interpreting the findings, especially when we are comparing our findings to other regions. For example, decline in species richness due to acidification can be more subtle in this region compared to Sudbury lakes, because these systems are already species poor. Therefore, our findings provide baselines to assess future impacts in this region. We also explored the potential of using indicator species to characterize acid stress levels. So here the idea is to identify indicator species to characterize different habitat categories. So basically, if you find that indicator species, you should be in that habitat type. And if you are in that habitat type, you should find that species. So Dufresne and Legendre in 1997 came up with an elegant method to identify indicator species based on their niche breadth. So they proposed to estimate indicator value for species by combining measures of specificity and fidelity. So specificity is highest when the species is present in a given group only, but not elsewhere. And the fidelity is highest when the species is present in all sites of that target group. I'll walk you through this idea by using a simple example. So here we are comparing two groups of sites which contain 4 different species. So we can see that species A is present in all sites of group 1, and they are they're only present in group 1. So therefore, they are appropriate indicators for group 1. On the other hand, species B is present in all sites across both groups. Therefore, they are not appropriate for either of group. Species C is only present in group 1, but they are not found in all sites of this group. On the other hand, species D is almost exclusive to group 2 and also found in all sites of this group. Therefore, they have a high indicator value for this category. However, this is a very idealistic and simplified example, and in reality, the data are more complex and messy, so we cannot identify indicator species by just visually examining the data. So Dufresne and Legendre came up with a statistical method to do this. So this picture gets more complex when we want to identify indicator species to characterize different levels in a stress gradient or a disturbance gradient. So we know that with the increase in stress, sensitive species will decline and they may be eventually completely lost at very high stress levels, as we can see with species D and species B. On the other hand, we can expect abundances of tolerant species that are usually generalists to increase with increase in stress, as we can see with species A here. So we can see that species D is an appropriate indicator to characterize low stress levels, but species A may not be quite appropriate to characterize highly stressed sites, because although we can see high abundance of them in these highly stressed sites, they seem to be omnipresent in almost all sites. On the other hand, species B is specifically absent from these highly stressed sites. Therefore, their absence can be more appropriate to characterize highly stressed sites. So we can define absence indicator species as species that are absent from all sites in a particular group, but are present in all sites of other groups. So we pursued this idea of identifying present and absence indicator species. So this slide shows the method that we used to estimate indicator value in each case. And we used a permutational procedure to determine statistical significance. So I should say here is that we are actually not using species presence absence data here. Rather, we are using abundance data to identify presence and absence indicator species. So we applied this approach to zooplankton data from Northwest Saskatchewan. So, and we considered ANC, or acid neutralizing capacity gradient, as a natural stress gradient. For highly sensitive category, we identified both presence and absence indicator species, and L minutus, the acid tolerant species I mentioned before, was identified as the tolerant species or the presence indicator species. However, its appropriateness is somewhat questionable because it's a generalist. We also identified several absence indicator species that are known to be acid sensitive species according to the literature. Therefore, we think a combined use of both presence and absence indicator species is appropriate to characterize these highly sensitive or highly stressed sites. We also identified both presence and absence indicator species for non-sensitive category. Again, these are in agreement with what we know about the ecology of these species from the literature. However, we did not identify any indicator species for this intermediate or sensitive category. So this means we can only identify indicator species to extremes in a stress gradient. So now I'm going to switch gears a bit and talk about a paleolimnological study where we look at temporal changes in lakes in northwest Saskatchewan over the past century. So this is actually a combination of two studies. So both of them are collaboration between University of Regina, Queen's University of Ontario, and provincial government in Saskatchewan. So one was conducted in 2010, where I performed the cadastral analysis component, and other one was conducted in 2014, where a colleague of mine, Brittany Hesadel, a master's student at UFR at that time, performed the cadastral analysis for her master's project. So we know that ecological responses to industrial deposition can be both a spatial and temporal phenomenon. So we know that industrial emissions have been increasing with increase in industrial production starting from 1980s. Therefore, we can expect ecological responses through time parallel to these changes. However, degree of response can vary spatially, as industrial deposition can decline with increase in distance from point source of emission. Also, climate in this region has been increasing over the past century. However, we don't see a clear pattern in precipitation, or at least we don't have adequate data to see a pattern. So ecological changes may be also happening in response to climate change. So let's assume we are comparing two lakes that are similar to each other with respect to acid sensitivity and other lake characteristics. So we can expect more or less synchronous changes through time in response to climatic forcing till intensification of oil sands production. However, they can be idiosynchronous after intensification of industrial activities due to varying levels of industrial deposition. So here we are assuming these two lakes are located at varying distance from industrial source. However, in reality, it's not that easy to find these similar lakes with respect to multiple criteria. At least that was the case for us. So the sediment cores were collected from each of these 14 lakes, and then they were sectioned into equal size intervals, and which represent chronological time periods. Then we identified and counted cladoceran remains belonging to different taxa in each of these segments, and this helped us to construct changes in cladoceran species composition through time in each lake. Then we wanted to model temporal changes in species composition through time. So first we summarize the major gradients in species compositional changes using principal curves analysis. So principal curves works better compared to conventional method like principal component analysis when there's a single gradient or one major gradient in the data. So this generated a single composite variable for each lake. Then we model the temporal trend in this composite variable or principal curve scores using generalized additive models or GAMs. So GAMs are appropriate to model nonlinear trends in the data. So we did it for all 14 lakes. To facilitate the interpretation, I'm going to divide the results into two sections. So first we are going to look at the lakes with ANC greater than 100. So here this black line represents the fitted trend, and these blue thick sections represent periods with statistically significant changes, and the dotted line represents the 95% confidence interval. So we can see that continuous significant changes through time in two lakes. We can also see temporal patterns are somewhat similar between 17J and 17V. So when we had a closer look at species data, we saw that these changes were related to increase in species that prefer more productive conditions. When we look at the remaining lakes with ANC below 75, we can see similar changes occurring in 4 lakes beginning around 1960s. So I should say here that the direction of change is not very meaningful here, and we are only interested in timing of change. So when we had a closer look at the species data, we saw that these changes were related to decrease in acid sensitive taxa and increase in acid tolerant taxa in 3 lakes. In contrast, it was the opposite in the other lake. So it was increase in acid sensitive taxa and decrease in acid tolerant taxa. It is unlikely that these changes are related to industrial deposition because they began well before intensification of industrial activities. Also, we don't see a pattern when we look at the spatial distribution of lakes with and without significant changes in 1960s. So we saw a significant change in 12D, although it's quite distant from the point source of emission. Although we did not see any change in 10W, although it is the closest to point source. So therefore, we think the responses we see here are related are influenced by climatic changes. So climatic changes can also influence acid-base status of lakes. For example, changes in runoff, downwater inputs, and water residence time can alter base cation concentration in lakes. Overall, our findings show that cladoceran communities are showing localized responses to regional climate change. And this is in line with this idea of filtering of climate change effects by landscape and in-lake filters. So finally, I'll talk a bit about spatial assessment we conducted in northeastern Alberta in collaboration with Environment and Climate Change Canada. So this study design consisted of 5 physiographic subregions with varying distance from industrial region. First, we ignored these subregions and look at environment-species relationships at a regional scale to see what factors are driving the species composition. Then we did the same again at sub-regional scale. So again, similar to our Saskatchewan study, we identified significant correlation between species composition and environmental gradients vulnerable to industrial deposition. Also, we saw some intriguing patterns in subregions closer to industrial region. However, we could not determine if they were related to industrial deposition or natural variability due to unavailability of baseline data. However, today I would like to focus on a different aspect of this study. So, conventionally, environmental factors were considered to be sole determinants of species compositional changes across a region. Therefore, it was expected that changes in environmental conditions can be reflected by changes in community composition. So we can track the changes in environmental conditions. However, this view has changed due to later developments in meta-community paradigm. So according to this paradigm, zooplankton communities are not isolated entities. They are rather connected to each other by dispersal process. Therefore, it's a meta-community. So therefore, spatial variation in species composition in a region are influenced by both environmental factors and dispersal process. This means our ability to track environmental changes using community changes is contingent on influences of dispersal processes. So we used an indirect approach to estimate roles of environmental factors and dispersal process for species compositional variation. So first we quantified variation explained by significant environmental factors. Then we modeled all spatial patterns in species distribution using a spatial modeling approach. Then we combined them together by using variance partitioning. So in variance partitioning, we can see what are the unique and shared fractions explained by each variable group. So this shared fraction represent spatial patterns in species data induced by spatially structured environmental factors. On the other hand, these unique or pure spatial factors can be attributed to dispersal effects. So at the sub-regional scale, we can see that environmental factors are more important and spatial factors are not significant in all cases. Therefore, we can effectively use zooplankton communities to track environmental changes at sub-regional scale. And at the regional scale, we again see that environmental factors are still more important. However, the spatial structures, although they are less important, they are statistically significant. So this means we can still use zooplankton communities to track environmental changes at regional scale, but we need to be cautious when we interpret species pattern in relation to industrial effects because community responses can be partially suppressed by dispersal processes. So as I mentioned, the approach we used to estimate role of dispersal was rather indirect. So there are several excellent studies out there that used more direct and robust method to estimate the dispersal effects of zooplankton. So I highly recommend these studies to anyone interested in learning more about this topic. So some of this work include work by Derek Gray and others looking at role of dispersal in recovery of zooplankton from acidification, and other one is led by Beatrix Weissner, who we have as a panelist today, looking at roles of environmental control versus dispersal on structuring zooplankton communities and other several taxa. So I'd like to end with few remarks. So it is important to consider natural variability, dispersal effects, and multiple stressors when we are using biological indicators. And I think use of new indicator metrics such as functional diversity indices are gaining momentum at the moment in the field of ecological indicators. However, I think their potential has not been fully explored with respect to using zooplankton as indicators. Also, there are a lot of new developments happening in the field of quantitative ecology, and I see a lot of promise in integrating these approaches with indicator development. With that, thank you for your attention, and I'd be happy to answer any questions.
Great, thank you very much. Just so I can look at people, I have the multiple screens going, so I think it's better for me to actually look into the camera. So I'll just fix my view here so I can see the chat box. I don't see any questions just yet. So I think in the meantime, while people are ruminating over what they've just watched and learned, I guess I could pose a question to all of the panelists, including the speaker, because I think what was really interesting is the kind of how things have changed over, say, the last 20 years with respect to the role of dispersal, the role of environmental factors. And I guess I'd like to ask, I'll start with Dr. Beisner, because your paper was cited there from 2006. And I know that you've been working in this topic prior to that. So I guess just give me your overall perspective on how things have changed and what we've learned.
Yeah, I definitely think we've started to consider more of the role of dispersal in recovery, especially. So Dr. Anas also cited Shelley Arnott's work, and I believe Shelley's here, and her lab's work, where they've been doing a lot with respect to acid recovery and the role of dispersal in that, which has been a really elegant demonstration of how important that is. Otherwise, I think a lot of the work, of course, we all know it's difficult to measure dispersal. So I think this is, of course, the biggest hindrance to advancing in how important that aspect is. So I think a lot of what we can say about is all inference, right? It's all inferring based on distances between lakes or water course distances and things like that, which is very unsatisfying. But at the same time, we know there have been some nice studies where they've taken, for example, empty mesocosms and set them up at different distances from lakes. This was, probably early 2000s. Carla Casares's group did quite a lot of this work, where they then looked at colonization of those tanks, for example, and showed that there was effects of distances from a lake and effect of rainfall and things like that. So we know it's happening. It's very unsatisfying, in terms of actually getting a real handle on how important it is, and we have to keep doing the statistical inference, which works to some extent, but it's not as as confirmatory as doing the analysis with environmental data, for example. So yeah, we can take it into account, but it's inference.
Interesting. Dr. Vissel.
I guess I would just add to that, I mean, while you say it's frustrating that we can't get that good of a grip on it, finding effects in